So in today's lesson, we're going to be learning a new topic, and that topic is meiosis. And so what is meiosis, you might be thinking? Well, if we take cell division as just like a general topic, so far, well, what have we learned? Well, so far we've learned about how the process of the nucleus dividing, we call that mitosis. And of course, there's interphase and cytokinesis along with that. But mitosis, when we're talking about how regular body cells divide, and then that happens via the process of mitosis. So, in other words, this is happening for somatic cells, which are your everyday body cells. Meiosis, on the other hand, I want you to take a guess. So meiosis is the process by which our gametes, or our sex cells, divide. Okay, so a couple things that you will remember from the last unit, hopefully, is that somatic cells contain the diploid number of chromosomes, whereas our gametes contain the haploid number. And that's because when our two parents' gametes come together, haploid plus haploid, so I'm going to call this 2n, and I'm going to call this n. So when you were born, well, when you were conceived, right, you would have had a haploid cell from one parent and a haploid cell from another parent to give you a diploid number of chromosomes when you were first conceived, when fertilization happened. So that is it. That's what we're learning today. Now, I think you'll find that meiosis is very similar to mitosis in terms of how the process works, but there are some differences, and they're very important differences, and so I'm going to start that conversation today. Okay, so first off, as I mentioned, the process of forming gametes is called meiosis. That's what it is. And in humans, it's going to take place, of course, if you're male in the testes, and if you're female in the ovaries. So again, we've talked about this, but feel free to include this in your notes in some way if you want to. In meiosis, as opposed to mitosis, the number of chromosomes in the daughter cell is going to be half that of the parent cell. So, this is a process that I'm going to explain to you all about in a short while, and I'm going to show you a visual that I think will help you to make sense of this. Okay, so we know that all gametes have to end up as being haploid. So how exactly does that work if you're starting with a diploid cell? And you have to, your chromosomes have to divide and such. So we'll take a look at that and try to make sense of it. So I thought I'd start with a diagram of mitosis and meiosis, just to compare them side by side, and so that you can see how they differ. So first of all, what I'm hoping you notice is that meiosis involves two separate divisions. And so that's really important, because we know that although we start with a diploid number of chromosomes in the parent cell, here's my parent cell, each of the daughter cells has to have 23, or the haploid number of chromosomes. So whereas with mitosis, our parent cell will have 46 chromosomes, but so will each of the daughter cells. And so you might be thinking, well, how does all of this work? So I'm going to try to explain that to you by drawing some additional diagrams, and hopefully this will make sense. So if I'm talking about mitosis, I'm going to be starting with a parent cell that would have an interphase. Before the end of interphase, just have a single, each of their 46 chromosomes would be, on their own. And then at the end of interphase, we would have that parent cell duplicate its DNA. And so in that case, what I have are, in the first case, I would have 46 singles, I'm going to call them. But then once interphase happens, the end of interphase, we're going to see that every chromosome is going to double. And so we have 46 doubles, if that makes sense. I'm only drawing one just for simplicity's sake. Right? But then, of course, we know what happens during anaphase is that centromere, which is in the middle here, is attached to spindle fibers, and each of those, that double gets broken apart. And so what happens is I end up with each of those sister chromatids just being each in their own. So, when I think about this 46 double right here, in total, that's just going to be 96 chromatids. Right? And so that will allow me to have 46 here and then 46 here, each of them being single, right? Because the 96 is going to divide by 2, and that's going to give me 46 chromatids in each cell, in each daughter cell. Now take a minute to digest that, because that's something in and of itself, before we move on to meiosis. Now let me show you how meiosis is going to work. So I know that eventually we're going to have to bring in the conversation around homologous chromosomes, which basically describes how really when we say in our 46 chromosomes that we have 23 pairs, right? So even though I haven't really depicted it here, we know that each of the 46 chromosomes is grouped. They're grouped into groups of 2 based on, there being 2 sets, one from mom, one from dad. So in order to better explain meiosis, that's how I'm going to begin. So I'm going to begin by talking about how we would have that. We would have a cell, and there would be, 46 singles. But now I'm going to say they're groups of 2. Why groups of 2 again? Because remember, you have a set from mom and a set from dad. So I'm going to color them differently. I'm going to color, let's say this one here and this one here, showing that these are representing a homologous pair. So I'm going to write that in, homologous pair. And they're starting off as singles. And so what happens in the first stage of meiosis is, this first step here, well, there's a number of things, but we can simplify it by thinking, okay, well, in this first part, what I'm going to see is just like I did in mitosis, I'm going to see each of these chromosomes duplicate. So that now instead, well, let me do my chromosome count. I still have 46 chromosomes in total, but I have 46 doubles instead. Right? So I also have, in this case, 96 chromatids. Let's keep count of that too. Okay, so I start off with, 46 singles, just like in mitosis. I'm depicting them in pairs here because we're talking about gametes, and that's going to be important. So in other words, 46 singles, 23 groups of 2, essentially grouped in homologous pairs. Then, at the end of interphase, we're going to see that they're going to the DNA is going to duplicate again. So that's going to give us still 46 pieces. It's just that the pieces are each individual piece has 2 components to it, 2 chromatids. So a total of 96 chromatids that we're working with. Let's keep going. So now, the first meiotic division, that is what we're talking about in this first part right here. So we've gone from here. I'm about to talk about what's going on here in that first meiotic division. So I'm going to show that. I want you to take a guess. What do you think is going to happen here? I'm going to end up with 2 cells. What's going to happen is one of those chromosomes one of the chromosomes in the homologous pair is going to move to one cell, and the other chromosome in the homologous pair is going to move to the other cell. So already what I've done is I no longer have 46 doubles in each cell. What do I have now? Well, I have 23, but they're 23 doubles, right, in each cell. Right? So that's going to give me, in each of these cells, I'm going to have 23 doubles, which means 46 chromatids. In each of these guys. Now I'm going to have to make this, I think, a little bit smaller to fit everything in nice and neatly, so bear with me here. I'll just make this a little bit. And so then what's going to happen from here? Well, I have to divide them again because, you know, I still have 46 chromatids. I can't have that. I need to have 23 single-stranded chromosomes to start off with for my gametes. So I need to see another division happen. So what's going to happen to each of these here, and I'm only going to show it for, well, I guess I can. I'm going to try to fit it in. Let me make this a little smaller. What I'm going to see is that for this, I'm going to end up with basically what happens in mitosis. The centromere is going to split. Those chromatids are going to move to either end of the pole of the original cell, and then cytokinesis will occur so that I have a single chromatid in each individual cell. So the same is going to happen over here with my blue chromosome originating from my homologous pair. And so that is how I end up with 4 times 23 singles. And this is how meiosis works. So I've backed up my note a little bit, and so what we were missing from before that you may want to jot down is this little bit. So in order to end up with cells that are haploid at the end of it all, the cells have to divide twice. So we consider the first division, we call that meiosis 1, and the second division, we call that meiosis 2. So let's do another recap of what I just presented to you about meiosis, because there is a lot there. This would consolidate it, and the diagram's a lot nicer. Let's go through it. So during interphase, we're going to have normally our chromatin, and once there's a signal for the cell to divide, we know that before that our DNA has to replicate. So we're just showing each. So we would have 22 other pairs just like this, but we're showing one for simplicity. Each of these chromosomes, singles, in the homologous pair is going to have to duplicate itself. That's why we end up with that standard chromosome. Now, each of the chromosomes is composed of 2 sister chromatids joined at with the centromere, right? So we have 2 of them. So, but really we have 4 singles grouped in groups of 2. So then they're going to each go to their own pole in meiosis 1. So in meiosis 1, the job is essentially for the homologous pairs to separate each into their own little cell. Then we're ready to start meiosis 2. So in meiosis 2, meiosis 2 looks a lot like mitosis in that what we're going to see is the spindle fibers are going to attach to our centromere and split these 2 sister chromatids in half, each going to their own cell. So since that happens for each of the chromosomes originally in that homologous pair, we end up with 4 overall haploid cells. So that was a lot to digest for a Monday, or for any day for that matter. So what I'm going to do now is I'm going to suggest that all you do is some follow-up work by looking at these homework questions in your textbook, numbers 1 to 3 and 6, and reach out to me for any questions if you have them. This was a lot. And if you require any clarification, I'll be happy to work with you to help support that.